Our program today will begin in just a few minutes. Today's program will begin in about 3 minutes. Today's program will begin in about 2 minutes. Welcome, everyone. We'll get started in just about another minute. Well, good morning and good afternoon to some of you on the East Coast. My name is Betsy Stewart, and I'm the Director of Advancement Communications here at the Leukemia and Lymphoma Society. And welcome to LLS Research Now, LLS's venture philanthropy model for accelerating promising therapies. Before we get started today, I just wanted to share a few housekeeping items. Please note that this call is being recorded. All participants' audio and video is currently disabled in order to cut down on background noise. At the end of today's panel, there will be a Q&A portion, an opportunity for you to ask your questions. You can ask questions during that time by typing into the Q&A feature in the Zoom. On your computer, you'll find Q&A at the bottom of your screen. And on a phone or a tablet, just tap the More button at the bottom of the screen on the right side, then hit Q&A. And if for some reason we don't get to your questions during today's call, we'll send the answer in a follow-up email. That takes care of all of our housekeeping items for today. Now I'd like to turn it over to our host for today's call, LLS President and Chief Executive Officer, Dr. Luis DeGennaro.
Thank you very much, Betsy. Hello, everyone. I'm Dr. Lou DeGennaro, President and CEO of the Leukemia and Lymphoma Society. And I'm truly pleased to welcome you all to LLS Research Now. This is the fourth in our new monthly series about the impact of LLS-funded research and how your support makes it all happen. Frankly, we really could not advance the science and the medicine as we have without you and your support. Today, our topic is the Therapy Acceleration Program, or TAP for short. And it's one of the most exciting and impactful ways that donors can influence the future of medicine through their support of the Leukemia and Lymphoma Society. As the name suggests, through TAP, we accelerate the development of new therapies for patients with blood cancer. We do this through a form of venture philanthropy, whereby we identify highly promising experimental therapies that are in the translational research pipeline, but they need an infusion of capital to advance the research that will help validate their benefit to patients. This translational research funding is critical, and let me tell you why. In medical science, there's a stage of the research continuum often called the Valley of Death. It's called this because the funding challenges usually faced by researchers when trying to move an idea ahead from the laboratory to the clinic can be quite daunting. This is the stage between when a new therapy idea has been validated in the laboratory and when it has been tested in the clinic. And it can be one of the most difficult to fund. You know, government grants from the National Institutes of Health and the National Cancer Institute are often available for scientific discovery, the early phase in the laboratory. And once an idea has been validated in the clinic, big pharmaceutical companies will be interested. But there's a gap in between the lab and the clinic. That's where TAP comes in. There are far too many extremely promising treatments that falter, frankly, due to this Valley of Death funding gap. Some of them could be potentially life-saving treatments. Since at LLS, our mission is to save lives, we do everything we possibly can to deliver better treatments and new cures to patients. In 2007, we launched TAP to help fill this funding gap and deliver those cures. We do it in partnership with supporters like you all. Every year, LLS reviews as many as 80 projects from biomedical research companies. We invest in only 2 or 3 new TAP projects every year, those with the highest potential to change the standard of care for patients with blood cancer. What's important about these projects, or one of the things that's important about these projects, is that they're much closer to impact for patients, only perhaps 3 to 5 years from FDA approval, when compared with projects at earlier stages of research. We're here to help lives. And that's what we do through innovations like the TAP program. And donors like you make this possible. Since starting TAP in 2007, LLS has invested more than $125 million and supported over 60 projects. 3 of these therapies, supported by LLS and our donors, have received FDA approval. That's actually quite an amazing achievement. One of those new therapies, and I'm sure you're all somewhat familiar with this therapy, it's a revolutionary chimeric antigen receptor T-cell therapy, or CAR T-cell therapy, is known by the trade name Yescarta. And you know what, folks? It is saving lives right now. Now, I'll be turning this call over to my colleague, Dr. Lee Greenberger, in a moment, but I wanted to take a moment to thank you all once again for your support, especially as we continue to deal with so much challenge and uncertainty in our world. LLS has remained focused on our mission to advance cures and support patients, and we do so thanks to our remarkable partners across the country and all of you on this call today who share our conviction that a future without blood cancers is achievable and worth fighting for. And with that, I'd like to turn the call over to Dr. Lee Greenberger, and he'll lead the conversation with our guests. Lee, over to you.
Thank you, Lou. My name is Lee Greenberger. I'm the Chief Scientific Officer at LLS. And let me just start my video so you can actually see me. Thank you. So TAP is a very exciting program because of the unusual nature, particularly because it's coming from a nonprofit, to move drugs into the clinic. So it's really a game-changing approach of which only a few nonprofits have in their arsenal. And it's through TAP, LLS, and the donors who are really at the vanguard of the most cutting-edge therapy. And by the way, TAP really selects first-in-class assets that have their high risk, but you'll see that they have actually impact patients, made it to FDA approval, and are saving lives. So while LLS supports much of the academic grants, and we have a very large grant portfolio, 250 grants placed worldwide in academic settings, there's no question that the pharmaceutical industry and the biotech industry are a very important piece of making sure that what comes out of academic labs actually can make it to patients. And we have some incredibly talented people on the phone who are going to tell you about the transition of academia over to biotechnology company, to large pharma, to patients. And all of these that you're going to hear from are bold thinkers looking for first-in-class assets, new assets that have never been tried before, to really advance therapies for patients. And it's really, truly a remarkable time to date that we've had so many drug approvals, and the advancement has been really quite remarkable. In addition to the 3 approvals that the TAP has supported, we also recently have supported the 47 Corporation, which was recently acquired by Gilead. And more importantly, they are developing a breakthrough therapy for myelodysplastic syndrome that looks amazingly promising. I want to point out and recognize a few people in the TAP program who are important in making this happen. 2 of them are Dana Callow, who is with Millennium Partners, and Casey Cunningham with Sante Ventures. And these 2 individuals have been part of our oversight committee and have led the oversight committee in this 12-year interval as we've moved many drugs into the clinic. And it wasn't for their valuable input and the input of the rest of the TAP committee, that's an oversight committee of what the internal folks at LLS choose. If it wasn't for them, we wouldn't be able to actually complete the mission of TAP and get the success we have today. So I want to recognize those 2 individuals in particular. I also want to call out the internal team within TAP. I work with Laura Grunberg, Blaine Robinson, Chun Zhu, and Javid Farazan to identify these opportunities, bring them to the TAP committee, have LLS fund these, and work with these companies. And finally, I just want to mention on December 4th at noontime Eastern Standard, there will be another TAP program available to you folks. It's called Value Creation for Companies through LLS TAP, and it's going to coincide with the ASH meeting coming up in December. Needless to say, and lest I forget, this work could not happen without your contribution. As Lou said, we're not government funded. This is all private money that we put to work. And if it wasn't for you folks on the phone and many of your compatriots, the patients, the caregivers, the entire community, this could not happen without you. So now I'd like to ask the panelists to turn on their video and microphones, and we can get started introducing the panel members and begin with the question and answers. So my first guest is Dr. David Chang. He is the Chief Executive Officer of Allogene. This is a relatively new company in the immunotherapy space, and I'll let him introduce himself in a moment. David was involved with LLS actually from a former company, Kite Pharma, and Kite Pharma, as Lou already alluded to, is the founder of Yescarta. So, David, could you please just introduce yourself briefly, and then I will guide you through some of the questions.
Sure. First of all, Lee, thank you very much for giving me an opportunity to be on this panel. I always viewed LLS as one of the most successful organization in the leukemia lymphoma space, especially bridging the private public sector. And I think everybody in this call who have supported LLS over a period of time should be really proud of what the LLS has done. So my name is David Chang. I'm currently the CEO and co-founder of a company called Allogene, which is focused on advancing the allogeneic chimeric antigen receptor therapy. This is a cell therapy that everybody thought was more in terms of belonging in science fiction than in the reality. But much to the, through the help of LLS, supporting the company that I was involved in early on in 2014, that is Kite Pharma, as we were introducing the first chimeric antigen receptor therapy for the treatment of non-Hodgkin's lymphoma. The product is now marketed as Yescarta, and it's one of the sort of most successful experience, for any drug development. And certainly, I'm benefiting from that experience by, moving from one company to now head up a different company with a different purpose, again, in the cellular therapy for the benefit of patients with cancer.
Thank you. I should point out that LLS started funding this type of therapy about 25 years ago, when the goal was simply to learn how to take these T cells out of patients and make them grow in tissue culture. That was among our first grants, and we have supported this through the grants. We continue to support that through academic grants, but it clearly was the companies that actually have translated this into the benefit for now about 3,000 patients who have been treated with Yescarta. So, Dr. Chang, could you tell us why the company that you were with at that time, Kite, why did Kite choose to get involved with TAP, and how has it been helpful for Kite as well as the whole CAR-T industry, if you will?
Yeah, so, as Lee has said, cell therapy has been around for some time, but that usually, most of the work was going on in the academic setting. It's, what the industry would consider as a one-off experience, where, the laboratory experience didn't really advance to something that could potentially be used in a broader patient population. So, that was much of, 80s and 90s, and, 2000 is when there appeared to be some major breakthroughs in the academic setting, that people were paying more attention. And really, even for somebody in the drug industry, the chimeric antigen receptor, that term was not really, widely used, or people knew what it was until some seminal papers that came out of University of Pennsylvania in 2011 and 2012 timeframe. So that's more or less the setting when new companies were being created. Kite Pharma was one of them. And Kite at the time was involved in advancing the CD19 chimeric antigen receptor program known as KTC19 at the time. So, I joined Kite in 2014 to oversee the research and development and also as a chief medical officer. So the dilemma that we were facing at the time was, all right, all the signs of chimeric antigen receptor therapy, this is an innovative therapy, very early signs of breakthrough therapy, benefit potential, meaning that the kind of treatment effect that we were seeing in some limited number of patients, this is something as a trained hematology oncology as myself, I had never seen such a, dramatic response, even in patients with refractory cancer. So that was really new. However, there were many challenges. One, this is a cellular therapy, which by itself, most pharmaceutical companies or industry would say that's not possible. There has not been any cellular therapy that's out there. Not to mention that the cellular therapy requires gene engineering. So this is a gene engineering cellular therapy that requires manufacturing. Patient by patient basis. And also, yes, the data looks pretty good, but this is all coming from, academic institutions. There is no real data that such a breakthrough, treatment efficacy can be reproduced in a large clinical setting. So, this dilemma of, something that had so much potential, but at the same time, there were so many barriers about how we can advance the program. And also, not to mention, 80–90% of the physicians out there have never heard about chimeric antigen receptor therapy, and this is totally new. So that was, the situation that we were in, and that led initially, our approaching Leukemia Lymphoma Society to help us advancing the awareness of the chimeric antigen receptor therapy and the cell therapy, because one, as with any new therapy, there needed education from a credible, unbiased organization such as LLS, and we thought that to be very critical. And that's when Lee and I started having a conversation, and Lee made me aware of this incredible opportunity of TAP program, where not only we can work with Leukemia Lymphoma Society, but we can also get some funding support for our clinical program. So when I learned that opportunity, it was a win-win situation. Being able to work with LLS to increase the general awareness of this innovative therapy that is on the horizon, and also being able to get the funding support. In many ways, the field will view this as somewhat of an independent validation. That's how credible LLS is, and any program or the activity that LLS supports. And this is 25 years of proven track record of unbiased, patient-focused activity that LLS has been involved in, and that really gave what Kite at the time would consider as too good to pass up. So we worked hard to get our program approved for the TAP. And we were fortunate that from the beginning of the program to getting the product approved, we were able to do it in a very accelerated time frame. From IND to the approval, that was about 3 years. And the outcome of that is that we now have a product called Yescarta, which has been on the market, and it's undergoing label extension from, more wider use from the initial use in the refractory large B-cell lymphoma. I'm no longer with Kite Pharma, I moved on, but the Kite Pharma has recently been talking about extending the indication to include a follicular lymphoma. And also with the same product, but under different name, they also achieved a approval for the use in the mantle cell lymphoma. So we are not Kite wasn't the only one. Novartis also was able to advance their product Chimera. And as we sit in 2020, we are potentially looking at rapid growth of chimeric antigen receptor product as other products are being reviewed for the commercial use. So, that was, in my view, one of the, greatest partnership. I hope, Lee, you feel the same way. Certainly, Kite always felt that was one of the best partnership that we were involved in.
Absolutely, I agree. I should point out to the audience that we not only contributed access to patients, and we provided financial support, but with the success of Kite and Yescarta, we also get some financial benefits from that back to LLS, which we put back into the mission. So it's a great philanthropy for us, and the reason why it's called venture philanthropy. Dr. Chang, I want to turn towards your new company and the future of CAR-T, and I want to explore where the field is going and why Allogene is the next step into making this more available and holds great promise.
So one thing that we learned during our Kite Pharma experience is, the current, treatment, Yescarta, it is what the field calls as autologous CAR-T therapy, which means that we need individual patients, the patient's own immune cells that needs to be taken out and that have to undergo manufacturing process to get the engineered cell, Yescarta, in this case. However, that can only be used in a single patient. It's a one at a time treatment. And also because it's a cellular therapy, there's a very complex logistics that's involved in collecting the immune cells from the patients, subjecting to a complex manufacturing process and ensuring that the right cell, there can only be one product that can go back to the patient, reaches the patient. So we learned, that there are a few things that could improve, and came to a conclusion that for the cell therapy to have continued success and really provide additional innovation to improve the patient benefit, the field has to move from this one-off autologous cell therapy to something that, one manufacturing process can lead to products that can be used in multiple patients. We call this as democratizing the cellular therapy. The benefit that we are trying to achieve is a greater access to the patient for the cell products, potentially controlling the cost of the cellular therapy, which has been pretty widely publicized in the media. And then also, improving the speed and on which the cell therapy can be made available. And, lastly, allogeneic cell therapy, because the manufacturing is done independent of the patient's need. We can do a lot more complex manufacturing, gene engineering and editing. And this is where the future of cell therapy, where, excuse me, innovation really comes in. So, that's what we are trying to achieve: access, cost, speed and reliability, and innovation. And that's where, the main purpose of creating Allogene. And currently we are advancing several different products, starting with one that's being developed for the non-Hodgkin's lymphoma, another one for multiple myeloma, and we will soon have a program for the acute myelogenous leukemia as well. So that's probably next 3 to 5 activities at Allogene, trying to advance the cellular products that can be used, that can reach more patients with the heme malignancies.
Thank you, David. We're going to move on now. Going to move from the West Coast, which is where Dr. Chang is located, to halfway across the country to Michigan. And I'd like to introduce you to Dr. Yolanda Grambecker. So if Yolanda, if you could bring up your screen and microphone, I'd like to introduce Dr. Grambeka. And Dr. Grambeka, we got involved with Dr. Grambeka, that is LLS got involved with Dr. Grambeka way back when she was actually at the University of Virginia and has developed, attempted to drug the undruggable, which I'll have to tell you about in a moment. So Yolanda, can you just introduce yourself first, and then I want you to talk about how the interface has been between academia, where you are at University of Michigan, and how that product has moved to market and how we've helped.
Thank you, Lee, for inviting me to this panel. This is a great pleasure for me to participate today. I'm associate professor in the Department of Pathology at the University of Michigan. So the research in my laboratory is focused on development of new small molecule-based therapeutics for blood cancer, with a particular focus on pediatric leukemias with unmet medical needs. So my laboratory is pursuing preclinical development of those new small molecules-based therapeutics using highly interdisciplinary research, including a lot of chemical, biochemical, structural biology, pharmacology, biological studies. So we try to incorporate all these components which are needed for the preclinical discovery and development of new therapeutics. So the project which I would like to discuss today, and that was really heavily supported by Leukemia and Lymphoma Society in the form of classical research grant approaches and also by therapy acceleration program, is the development of new therapeutics to block the oncogenic activity of the oncogenic protein called MLL, mixed lineage leukemia, which is particularly important in the aggressive subtype of leukemias with translocations of the MLL gene. So this leukemia subtype occurs in about 10% of all acute leukemia patients, both AML and ALL. But it's particularly prevalent in infant leukemias because it constitutes about 80% of all acute leukemia cases in infants. So my laboratory has pioneered development of small molecules that inhibit the protein-protein interactions between this MLL oncogenic protein and another protein called menin. So that's what Lee has mentioned, using this undruggable approach, because at the time when we started the project almost a decade ago, the blocking protein-protein interactions was considered like a very challenging goal. Sometimes these interactions were considered undruggable. But we decided to take the risk, and also being in academia gives us some advantage of, maybe tackling more risky projects initially, de-risk it, and then maybe move forward with the partnership with the company. And that's exactly what we did for this particular project. So as Lee has mentioned, we initiated this project when I was still at the University of Virginia, 2008, 2009. And then actually Leukemia and Lymphoma Society supported the very initial stage of this project with the TRP grant. That was the screening for small molecules to block this particular protein-protein interaction. And then for the subsequent several years between 2012 and 2016, we were supported by the TAP program. And again, it was very unique probably for LLS too, because I haven't seen many examples of the preclinical development in academia in particular to be supported by TAP. So we feel really privileged and really fortunate to be able to be supported by this program. So basically, as I mentioned, LLS played a critical role in all stages of developing these molecules, and especially the TAP program has helped us to go successfully through this valley of death, where you have to be very effectively and extensively move forward to prepare, make, and test lots of molecules. In our case, it was more than 1,000 compounds that we synthesized and tested in my laboratory. So we had to have a team of people that could possibly, go through all these stages, and eventually we are able to identify very effective small molecules which and demonstrate for the first time that we can block progression of the leukemia in mouse models. So at that time, we got really attention from the industry and decided to partner with Cura Oncology to advance our compounds to clinical trials in leukemia patients. And indeed, for the subsequent 3 years or so, we still were working, my group worked closely with the Cura Oncology, and we developed this molecule KO-539 as a co-developed with Cura. And this compound is currently in phase 1 clinical trials in AML patients. So the trial has started about a year ago, and we are going to hear the results of the clinical studies in about 6 weeks at the ASH meeting in December. So we are very excited and very eager to learn what happens in patients, but it has been really a great journey, and it only was possible due to the support of the Leukemia and Lymphoma Society. As I mentioned, these molecules are very promising. So in mouse models, especially KO-539 compound induces complete and long-lasting remission. So we are hoping that we can see similar outcome in leukemia patients.
Thank you. I want the audience to understand it's basically been about 12 years to get this into the clinic. And this is basically a drug that initially was for a rare leukemia, right? High risk program, drug protein interaction, very difficult to do. Dr. Grambecka, you developed very potent inhibitors that look very interesting. Now somebody has to actually take the risk to develop this in a limited market. So I actually ran into the CEO of Cura Oncology, the company that took it over, and it was a fortunate thing because they got it. And they took additional risk. And so I want the audience to understand that the risk of doing the expensive work in terms of toxicology and doing the clinical trials and doing the logistics of the manufacturing, these are all expensive undertakings that a company can be prepared to do, and they're doing it. I know that you've gone on to go after other undruggable targets. Tell us a little bit about, about what you're doing now and why you're doing it to go after some additional work, in particular,
right,
the ASH molecules.
Right. So yes, in addition to the menin inhibitor project, which, as I said, is now in the clinical trials, although we still are doing lots of preclinical studies testing maybe other indications, and we found that other subtype of acute leukemia, like NPM1 mutated leukemias, are also sensitive to the treatment with our inhibitors. So that indeed expands the patient populations and potentially helps to expedite the clinical trials as well. And also more patients can benefit from this type of studies. So in addition to menin inhibitor, also combinations with other drugs, because even though we strongly believe our compound can be very effective as a single agent, in some cases it might be even more effective if we combine it with other targeted therapies, such as we just published a paper which came out in Blood a couple of weeks ago on combination of menin inhibitor with the FLT3 inhibitor, the kinase inhibitor, demonstrating indeed basically long-lasting remission. So that's menin project. In addition, in my lab, we have several other projects. They are all similar type, in most cases blocking protein-protein interactions. And one of the projects, which is also supported by the Leukemia and Lymphoma Society, is the inhibition of ASH1L. It's another protein heavily relevant to leukemia. So basically, ASH1L is a new epigenetic drug target, which induces methylation of histone proteins. And it's highly relevant to pediatric leukemias, not only with MLL translocations, that's one of the indications, but also we found that other subtypes of leukemia are dependent on ASH1L catalytic activity. So my laboratory has started development of small molecule inhibitors to block the catalytic activity of ASH1L. It's a histone methyltransferase, it's an enzyme. And this is supported by the LLS Screen to Lead program, and we work very closely with Leukemia and Lymphoma Society to perform the screening and then optimize the several classes of compounds. But also we are developing the degraders, so-called PROTAC molecules, so small molecule degraders of ASH1L, which would also demonstrate potential as a future therapeutics in patients. So this is supported by the LLS TRP grant program. So these programs focus on ASH1L blocking or degradation are still at early stages of preclinical development. but we are trying to follow up similar pathway as in case of menin inhibitor. So hopefully they look promising and we can achieve the similar outcome, meaning translation to the clinic at some point of our work.
That's your concept about degraders is a perfect introduction to the next speaker. Thank you very much. And I'd like to introduce Jared Golub from a company called Chimera that is studying these degraders. We recently, TAP has done a deal with Chimera to advance these molecules. So I think this is Jared, I want you to just introduce yourself, and then Yolanta mentioned degraders. I want you to talk a little bit about what the concept is with these degraders, why they're novel, and why they might be important.
Okay, great. Well, thank you, Lee, and thank you again for the opportunity to be part of this panel this afternoon. So my name is Jared Golub. I'm Chief Medical Officer at Chimera Therapeutics. Chimera was founded approximately 4 years ago and is a company which is really focused on developing drugs using what is called targeted protein degradation. So we're solely focused on that technology to develop novel therapies for a variety of different conditions, serious diseases, whether they be oncologic or inflammatory. We're somewhat disease agnostic. And with regards to what is targeted protein degradation, so cells, have their own endogenous mechanism for what we call protein homeostasis, for controlling the levels of proteins. And that includes not just the synthesis of proteins, but also the degradation of proteins as they grow older. And there's a system called the UPS system, or the ubiquitin proteasome system, that normally is around, which tags proteins that have become older with what's called ubiquitin, and it tags them for degradation by what's called the proteasome. So this is a normal system that cells use to degrade proteins. Targeted protein degradation actually hijacks or co-opts that system in order to degrade proteins of interest, proteins that are either disease-causing or disease-modifying. And we do that through the synthesis of compounds or molecules that we call heterobifunctional small molecules. And these molecules essentially have two different components that are put together with a linker. One component is a ligand that binds to a particular protein of interest that you want to degrade. The other part of the molecule binds to a component of that ubiquitin proteasome system called an E3 ligase that then brings that into proximity with the protein of interest so that that protein can get ubiquitinated and then degraded. So essentially, you're using the UPS system to go after and degrade a protein that might not normally be the target of that particular system or E3 ligase, but now becomes the target because you've added in this heterobifunctional small molecule. And this really allows us to potentially have a transformative technology where right now, with traditional small molecules, monoclonal antibodies, RNA therapeutics, perhaps we're able to drug, perhaps maybe 20% of the so-called proteome. So that leaves 80% of the proteome that still is undrugged. And many of these proteins are involved in either causing or modifying diseases. And so by using, targeted protein degradation, we can now open up potentially this other 80% of the proteome and allow us to go after targets, be they targets in oncology or in other sorts of diseases that were previously undruggable, and allow us an efficient mechanism to really go after these targets.
Okay, great, thanks. So why LLS? Why blood cancer? Why'd you come to us? Or vice versa?
Yeah, so we're obviously, we're very interested in developing degraders to go after targets that are either been poorly drugged or are undruggable. And it so happens that some of the targets that we have chosen as our first programs are targets that are parts of pathways that are integral, to certain types of cancers. So for example, our two programs that I'll discuss, one is targeting a protein called IRAK4, the other is targeting a transcription factor called STAT3. Both of these are involved in hematologic malignancies and are considered either poorly drugged or undruggable targets. So for example, IRAK4 is a protein that is involved with signaling through what is called the midosome. And the midosome, one of the proteins in the midosome is called MyD88. And it turns out that in diffuse large B-cell lymphoma, about 25% of these patients have a mutation in this MyD88 protein, making those cells dependent on IRAK4. So going after IRAK4, targeting IRAK4 can be an effective way to treat patients, 25% of patients with diffuse large B-cell lymphoma, especially patients who have failed or have progressed after multiple other therapies. On the other hand, STAT3 is a transcription factor that has never been successfully drugged. We now have a very potent, very selective degrader of STAT3, and we know that there are certain STAT3 dependent hematologic malignancies where there's a high unmet need, especially peripheral T-cell lymphoma, cutaneous T-cell lymphoma, large granular lymphocytic leukemia, and even certain subsets of diffuse large B-cell lymphoma. So we now have the tools to be able to drug these important targets and potentially treat these hematologic malignancies with high unmet need. But this requires having a very focused and intelligent approach to drug development and having the right collaborations and the right networks with patients and really creating awareness among both clinicians and patients. And so LLS really seemed to be the ideal partner for us, given its deep scientific expertise in hematologic malignancies, including lymphoma as well as leukemia. The fact that LLS can really advise us on our clinical development plan and give us both a scientific and clinical perspective. The fact that LLS already has connections with key academic collaborators and potential industry collaborators as well that we can plug into. And importantly, the fact that LLS is very well established and network within the actual patient community, be it lymphoma or leukemia. It can really help with making patients aware of these new therapeutics that we're developing, and also making their clinicians, their physicians also aware, and really allowing us to work together with both patients and physicians to develop these drugs in a timely manner and to bring these new therapies to patients.
Thank you. I want to amplify a little bit from the LLS perspective. So we have 250 active grants. We get 800 applications a year. We have just basically figured a hub that reaches out globally to many academics. And so we knew when you came forward and say you got an IRAK inhibitor, we knew that mid-D88, which is just a little bit farther upstream, if you will, or controller of IRAK, we knew that's a hotspot. And so we said that's something very important to us. And here was a company with the technology and the will to do it. So it really looked like a really good marriage for us, with benefit to all. I do want to move to the questions, but one of the questions, and I think this has come up in the last two speakers, one person's asking, what does undruggable mean? And I want to put that, the answer should come in the context of, we have these so-called enzyme inhibitors, which we do like, it's a piece of cake. So can somebody tell us what the difference between enzyme inhibitor, piece of cake, and undruggable means?
Yeah, it's a really good question. It's a really important question, and it's not as easy to answer as one might think it is. But I can try to give one answer to that question. So undruggable, if we think about a target for which we can hit it, based on current technology. So either a small molecule that's able to block the activity of that particular protein or the enzymatic activity, or a monoclonal antibody that can also block, the active site of a particular protein, or even an RNA therapeutic that can block the transcription and thus the translation of that particular protein. Those would be considered relatively druggable. But undruggable targets are those targets for which there either isn't anything which binds to that particular protein yet, because it's been very hard to find a so-called ligand, and transcription factors, for example, are classic examples of proteins that are very hard to find something that will bind to it, that can get you sort of a foothold onto that protein to allow you to modify its function or to modify its expression. In addition to the fact that there are proteins where you're not blocking its enzymatic activity per se, you're blocking other functions of that protein, even if that protein has a scaffolding function, where just the physical properties of that protein allow other proteins to come and bind to it that's involved with this activity. So drugs that are just inhibiting the kinase activity of a protein may not be affecting its scaffolding activity, for example. So in order to really affect both of those things, you have to get rid of the protein. That's an example of an undruggable protein, where, for example, only a degrader would be able to really help you, because the degrader is getting rid of the entire protein, not just trying to block its activity. These degraders also allow us to find these hard to find ligands to proteins where finding binders has been very difficult, but we just have to bind enough to the protein to allow it to come together with this degradation system and allow that protein to be ubiquitinated and degraded. So it opens up more opportunities for finding ligands that don't have to bind extremely tightly or bind to an active site of the protein. It just has to bind enough to bring it together with this degradation machinery to allow it to be degraded. So those are the sorts of undruggable proteins that we think we can really address with these protein degraders.
Thank you. So I'd like Yolanda and David, you can open up your mics and come back on. Because Yolanda, so we talked about a degrader. I want, why is a protein-protein interaction where two proteins come together, why is that so undruggable? Why can't we develop lots of molecules to block that interaction?
Right. I mean, obviously, within protein-protein interactions, there are different subcategories, but in general, the protein-protein interactions are considered undruggable because there is typically no well-defined pocket present at these interfaces. So the interfaces are flat, and there is multiple contacts between the proteins. So it's very difficult for the small molecule to mimic all these contacts from the binding partner to effectively inhibit this protein-protein interaction. Obviously, there are exceptions, and actually in case of menin, we found a fairly well-druggable pocket. It was actually a huge pocket, but in many protein-protein interfaces, there's lack of well-defined pockets. So that's why these interactions are much more challenging or sometimes undruggable, as opposed to the well-defined pockets typically present in the catalytic subunits or in enzymes.
Thank you. I'm going to move to the questions. I have one question that's asking, and Lou, I'm going to ask you to come back on if you're still there. Talk about, we're providing funds to companies. Companies go out and sell these products and they make profits. What's the benefit for LLS? I mean, what's the benefit for patients? What's the benefit financially for us? Why do we do, we're going to get an income from this. So how does that matter to us?
Well, so as you know, and it's actually been covered a couple of times here, I think LLS uses a selection process to select these company partners, much in the same way it uses to select the academic research institutions that it partners with. Very deep process of critical evaluation of the research program, and in the case of companies, not only of that, but of the financial status of the company and the management and so on. But everything is driven, first and foremost, by the desire to meet an unmet medical need. And the LLS funding in this case is distributed to these companies in the form of, we'll call it sponsored research, a sponsored research agreement, in a way not unlike when we give a grant to an academic institution. Now, we do contemplate as part of the agreement a potential financial return on investment to LLS. And Lee, as you mentioned, if we are so lucky as to see a financial return, those dollars are put immediately back into supporting our mission program. But I hope that helps answer the question. Thank you, Lee.
Mm-hmm. Dr. Chang, I want to take it back to immunotherapy. And clearly, CAR T therapy has transformed the blood cancer space. Can you tell us, put this in perspective. So the immunotherapy, certainly the checkpoints, and I think you should go ahead and define that in the solid tumor space. But what has been the influence of the progress in CAR T therapy in terms of immunotherapy in general and immunotherapy for cancer?
So, I think this is, where, the idea that has been around, for a long time, there was a period of what I would say, dark ages where simply dismissed. And, the whole idea is that, patients or your own immune cells can go after cancer, tumors. So, the whole idea of sort of changing, manipulating, that, important, the body's function and making that, your own immune cells to work against the tumor. I mean, that's really the, the basic foundation of, immunotherapy as we know. And, much to the, the seminal discoveries that has been made in so-called the checkpoint inhibitors, like anything in the body, there's a checks and balance. When we say checkpoint inhibitor, these are the, checks and balance that's in the body to make sure that the immune cells do not go crazy, because when that happens, it has its own negative consequences. That's the natural balance, which tumor cells seems to take advantage of to keep the immune cells away, from killing, eliminating the cancer cells. And the checkpoint inhibitor, which is one of the, probably the brightest new addition to how we are fighting the cancer, over the last 10 years. I mean, that sort of controls that, counters this checkpoint inhibitor to allow the immune cells to kill the cancer cells more effectively. So that's one. Another area that has really gotten a lot of attention is, well, that's one way, just like, protect and redirecting proteins for degradation, there's a concept of redirected T cell therapy. Here, what you're doing is trying to recruit the, T cells to the cancers. And this is where, chimeric antigen receptor, we frequently say what we do with the gene engineering in the chimeric antigen receptor therapy is essentially, putting a GPS to the T cells so that the T cells can identify tumor cells and kill the cells. I mean, that's the part of the, very simple view of what the gene engineering is. But that's another concept that's gotten a lot of attention, either as a chimeric antigen receptor therapy or as a bispecific T cell engager therapy. One example of approved product in that case is Blincyto, which is a CD19, CD3 T cell engager, bringing the T cells to the cancer cells. So that's really sort of the beginning of the immunotherapy. And those who are in the immunotherapy, and also I think anyone in the cancer treatment setting is really coming up with a specific ways to kill the cancer cells without causing many undue side effects. I mean, which I think is really what differentiates with what the current drug development in the cancer space is doing versus 20, 30 years ago, where the goal of the cancer treatment is give as much cytotoxic chemotherapy that you can give to kill the cancer cells without killing the patients. I think from that somewhat, nowadays I think people will say that's a really little bit barbaric approach, but at the time that was the only solution. But now with advancement of the science, there are many different ways that we can handle the cancer.
Thank you. So let's amplify on that a little bit. Certainly with COVID and vaccines being very much in the news, here's a question going like, so why can't we just develop vaccines for cancer? I mean, we do have for HPV, we have a vaccine. Why can't we just simply use the immune system and create vaccines and kill cancers that way?
Should I answer that question, or?
Anybody wants to go for it, but you seem probably kind of front and center for that.
No, I mean, the vaccine, is a preventive measures. I mean, vaccine has worked very well in preventing infections, or in the case of, HPV, preventing that infection to really establish in the body. If we can do the same for cancer, with antigens, so these are the proteins or things that will distinguish cancer cells from normal cells. With the right antigen, I think the vaccine therapy can work as a preventive therapy. However, when the cancer cell starts developing and starts growing, it's the horse is out of the barn. One of the fundamental feature of the cancer cells is they continue to mutate and change to overcome or find a way to bypass patient's immune system. So in that setting, you are going after target that changes constantly, and going after with a single vaccine approach will be a very challenging proposition. But if you really sort of think about what may happen in the future, I think for the society and for the patient, some kind of preventive measure is probably more effective way to combat cancer rather than trying to handle disease after it has spread throughout the body.
Thank you. Well, we're certainly looking at that. I see that we're down to like the last 4 minutes. I'd like to turn it back to Dr. Lou DeGennaro to close it up and make the final comments. So, Lou?
Lee, thank you very much. I want to express my sincere thanks to the panelists for the clarity they brought to the call and for the exciting work that they're doing on behalf of patients. Thank you. Thank you very much. Thank you for joining us. And Lee, for your, as always, constant guidance through the panel. To everyone on the call today, I can't begin to describe how pivotal your support is and how deeply we value your partnership, especially now as LLS, like so many other organizations and nonprofits, navigates the challenges of our current reality. And as you know, the LLS mission is even more important today for patients than it ever has been. I'm happy to announce that our next LLS Research Now call will take place Tuesday, November 17th. So watch for the announcement of that. The topic will be a very interesting one. The topic will be advances in prevention and survivorship research. Folks, I've been working in this area for 25 years. I have to be honest with you, I never would have imagined we'd be using the prevention word with respect to cancer research and cancer therapy development, but tremendous advances have happened, and we are moving programs forward that potentially can operate in the realm of prevention. So I think you'll find this to be an extremely interesting topic and will bring some, as we have today, some exceedingly talented scientists and physician scientists to the call. It'll be hosted by our Chief Medical Officer, Dr. Gwen Nichols, and of course, Dr. Greenberg will moderate the panel. So again, keep on the lookout for some additional information about that. So with that, I want to thank everyone who's joined the call today. As has been said multiple times today, we really cannot do this work without your help and your support. And with that, we'll call the call to an end. Thank you all. Have a good day.